Namaste students. Today our topic is single chain antibodies or single chain variable fragments. They are similar terms. Single chain variable fragments are the fragments actually which are created when variable regions of heavy chain and the light chain are combined. They are fused. Okay. What are the variable region? Variable region is the region which will determine the type of antibody. It will determine the nature of the antibody and it will differ from antigen to antigen. There are lots of antigens which are present in the environment, and for those millions of and billions of antigens, we need billions of antibodies. So only the variable region will be changed, not the whole antibody will be changed. For that, only variable region will determine the type of antibody. Okay, so on depending on these fundamentals, some antibodies are designed in which the variable regions of the antibodies are fused. Okay, so antibody have two portions, light chain and heavy chains. In the light chain there is a variable region, in the heavy chain there is a variable region. So the variable region of light chain and the heavy chains are combined and fused for the preparation of single chain antibody. Okay, there is short linker polypeptide of 10 to 25 amino acids which is known as the single chain antibody, and the linker is usually rich in glycine. Okay, the most amino acids are rich in the glycine region, which have N terminus and the C terminus of the VL region. So the variable region, as we know that it has the antigen binding sites. So this variable region only will be the bind to the antigen binding site, not the whole antibody. So we need to mix the two variable regions each other by the hybridoma technology. Okay, so this is the basic mechanism by which the antibodies, single chain antibodies are prepared. How the single chain antibodies are prepared and how they are purified. As we know that antibodies are single chain antibodies are in the FAB portion, and the whole antibody is in the FC region. So the variable region is separated by protein G. Okay, protein G. By this protein G, we can separate the antibody variable region, and then it can be further purified using the protein L. So these two proteins are used for the purification of the single chain variable fragment. How the single chain antibodies are prepared in the bivalent and the trivalent single chain variable fragments. They are, if one single variable fragment will bind to another single chain variable fragment, then it will create a divalent fragment. And if one divalent will mix together with a single chain variable region, then it will be a trivalent. So we can understand it by the diagram here. Uh, this is the antibody. This is the mm, single chain antibody, and the variable region of these two will bind each other, making it a di, making it a di single chain variable fragment. Okay, actually this is just a single, just This is just a for our simplicity, but actually it will look like it. VL will be it will be look like in the 3D formulations, and when these two will be combined by another single chain variable fragment, then it will be like this. Try single chain variable fragment like this. That will lead to the original structure. So what are the examples of the single chain variable fragments? This is the example: actually jump, and another is C6 1.5 targeting HER2, which is used in the breast cancer examinations. So there are various examples which are prepared in the laboratory conditions as a single chain variable fragments or single chain antibody. So our lecture is over here. Thank you very much.